Well, good morning, everyone. Good morning, Brazil. Good afternoon in Austria. I would like to introduce Professor Dr. Verena Dirsch. The professor works in the Center of Pharmaceutical Sciences at the University of Vienna. Her expertise are investigations with molecular targets and drug discovery from nature. Well, she has published many papers in the areas of pharmacology and pharmacognosy. So it's an honor that you have accepted our invitation to conduct a seminar for our students. Okay, now you are the presenter. Thank you, Professor Verena.
Thank you very much for this kind invitation and also for the very nice introduction. It's a pleasure to talk to Brazil. It's amazing that things like that work today. Yeah, I will talk today a little bit about nuclear receptors as targets for natural product and natural product analogs. And before I do this, I would like to introduce a little bit where I come from and how we are organized. So can you see my cursor?
Yes.
Okay. So as you see here, we are from the Department of Pharmaceutical Sciences, as mentioned already. And here in this department, we have 4 different divisions. I think the classical fields like pharmaceutical chemistry, technology, and biopharmaceutics, and pharmacology. And here the green leaf, this is the pharmacognosy. So we are in the division here of pharmacognosy. And you see here also the building, and we have 3 different labs in pharmacognosy. So on one side, we have more this classical area. The lab is called Phytochemistry and Biodiscovery. It's led by my colleague, Judith Rollinger. So what they do is you're doing isolation, structure elucidation, but they do it on a very high level, very sophisticated, using very nice techniques, also in silico tools, AI tools, and so on. In addition, they have also a model for Caenorhabditis elegans, so they can also on their side test their compounds. But very often they then come to us, which is nice. On the other side, we have another lab that's called Pharmaceutical Biotechnology. That's led by my colleague Sergei Tsuchev. And he's interested in natural products from microorganisms. So they are basically focused mainly on natural products from microorganisms and focus on antibiotics mainly because there's a huge need in antibiotics, and microbes are a major source of antibiotics still today. So my colleagues here on the right and on the left are more in the area of on the compound side. So they try to find new compounds, isolate new compounds. And we are in the middle, so we are calling our lab the Molecular Targets Lab. And we are more in the pharmacological area, so we're trying to find out what these natural products are doing. And also try to find molecular mechanisms and so on. And we're doing this actually quite for a long time now. So I'm here now for 21 years at the University of Vienna. And over the years, we have established quite some cellular models. So this just shows you a scheme trying to explain what we are doing in our lab. So basically, in the center, of course, are natural products, and so isolated natural products, but also extracts, for example, from plants. And so our area of interest in pharmacology is around cardiovascular, metabolic disease, and here in the center is always the inflammation. And over the years, we have established quite some cellular models for phenotype-based cellular models. So where you generate, or where you have a model and you can measure a specific phenotype, like, for example, we focused on vascular smooth muscle growth and migration because this is important in cardiovascular disease like atherosclerosis and restenosis, or the nitric oxide release from endothelial cells, which is very important for endothelial function, or if they are too less, for endothelial dysfunction. We have established a model that can measure the cholesterol efflux from macrophages, which is very important for the whole reverse cholesterol transport, so that cholesterol is transported back to the liver and finally excreted. A postdoc recently also established a new cellular model that can easily measure CMP accumulation. So here we're going more in the area of G protein coupled receptors also. And we have also worked with gut cells and tried here to measure or set up cholesterol uptake, but it turned out to be so complicated that, to be honest, we don't do this anymore. But the basic principle is that we try to see what if you test a compound in such a model to measure an effect, and then in the end we would like to know how does this effect occur, so what is the molecular mechanisms, what signal transduction is affected. Which can be, to be honest, quite tedious. So we were chasing also resveratrol for decades and trying to find out what it does, but we had been not successful and stopped finally. Today we are focusing more on target-oriented cell-based models. So here we use luciferase reporter gene models. So I will explain this later how this works. And with this, we can measure effects of natural products on transcription factors like NF-κB, NF2, NFAT, CREB, but also and especially on a lot of nuclear receptors. And I will come to this later in a little bit more detail. So if you have cellular models, of course, or cellular pharmacological models, what you need are compounds. So on one side, I explained already, we are taking over compounds from our colleagues in-house. But we have also, what you can see here on the left side, we had from 2008 to 2014, we had established a huge collaborative network in Austria, which included quite some universities. And this was a huge project that was funded by the Austrian Science Fund, and the title was Drugs from Nature Targeting Inflammation. And here we really tried to bundle the whole expertise available from the chemistry side, from the modeling side, from the pharmacological side to identify new natural products that have some sort of effect in the inflammatory process. And this database that we created in this consortium, we still do have. And it turns out that if you, for example, test now this database against new targets that have a role in inflammation, it's very productive database. So much more productive if you would use any database. So it's really, so it was optimized also this knowledge from folk medicine to optimize it for inflammation. On the other side here, on the right side, we have also in the meantime a collection in the pharmacognosy division. But if we, it's about 5,000 compounds, but this would be just random screening if we would do this. So normally we don't do this. But this DNTI database, we screen again and again with different targets, and we find always quite interesting new things. This is just to show you how productive specific areas are. I mean, on this endothelial nitric oxide project we've stopped working for quite some time. Also, vascular smooth muscle cells are not so interested anymore for us. So we published this year a paper, but overall, so this is not so much worked on anymore. And also here, macrophage cholesterol efflux, it was mainly done by a postdoc, and when he left, we also abandoned this area a little bit. But it's still an assay we can do. NF-κB activities is, always of interest. So from time to time, we have papers here. We were working also on TGF5 and some postdocs, like my colleague, she's not postdoc anymore, she's associate professor. She's working a lot on NF2, and a postdoc here, he established models for PDE inhibitors. But my current interest is mainly nuclear receptors. And here we are most active currently. And so this is the reason why I'm trying to focus now on this topic. Oops. So just as a repetition, what are nuclear receptors and how do they look like? How do they work? They are constructed always in the same way. So because they are receptors, we have a ligand binding domain, which is the blue part here. So the ligand binds. And so you have here on the left side, the primary structure, here the tertiary structure. And I would like to emphasize already here that in this ligand binding domain, you have an activation function, meaning that the ligand that binds can influence this activation function. On the other side, because these nuclear receptors work also as transcription factors, they have a DNA binding domain with which they bind to the DNA, and they have a second activation function. So these are molecules that can be regulated by ligands, and since they act as transcription factors, this ligand can directly influence the transcription of certain target genes. There are several types of nuclear receptors. I will explain this later, and they also have different ways of functioning. So I just try to explain here a little bit how this works. I mean, you probably are aware of this more classical steroid binding nuclear receptors, the steroid receptors, and they are sitting normally in a cytoplasm, and they are bound to heat shock proteins. And when now a ligand enters the cell, they bind to the ligand binding domain, and then these heat shock proteins are released and the nuclear receptors enter the nucleus. So this is the nucleus here. This is the cell membrane. And then in the case of this classical steroid hormone receptors, they build dimers. So they bind as dimers to the response element, and then they lead to or then the transcription of a target gene is activated when co-regulatory proteins are recruited. So it's not only the receptor itself, you need also co-regulatory proteins. And these co-regulatory proteins, they can suppress the transcriptional complex or activate it. So you need co-activators in order that bind to the whole complex in order to activate the target gene. Then there are also some receptors that reside already in the nucleus bound to the DNA. These are the so-called type 2 nuclear receptors, and typically for them is that they bind as heterodimers. So here the nuclear receptor, here the green part, builds a heterodimer normally with another nuclear receptor, the so-called RXR or retinoic X receptor. And these heterodimers are normally bound to co-repressors. So in this state, when there is bound a co-repressor, they are inactive and no transcription occurs. And when that ligand then binds, it can bind to the green part, to the transcription factor of the green part, and on the other side, also to RXR. Then the transcription starts. So the co-repressor is released, co-activators are recruited, and then the transcription starts. And here again, we discriminate between so-called permissive heterodimers and non-permissive heterodimers, because the permissive heterodimers, they can regulate it also by ligands for the retinoic X receptor, whereas the non-permissive one, they bind ligands for RXR, but they cannot co-regulate the whole receptor or the whole receptor complex. And then there on the other side are also receptors that act as monomers that bind ligands and regulate as monomers transcription. And here is something that is very, very important to understand. So nuclear receptors are not simple on and off switches. So this was initially, I think, the thought of researchers, of scientists. They thought, okay, here is a switch. I can just create a ligand as an agonist, for example, and then I can switch on, and as an antagonist, I can switch it off. But it turned out to be much more complex. So on one side, you have the ligand that binds to this ligand binding domain, and each specific ligand creates a very individual receptor conformational change. And this very individual conformational change creates very unique protein-protein interactions with your co-regulatory proteins. So not every agonist is the same, not every antagonist is the same. So it really depends on the structure of the ligand, how the conformation is changed, and how in the end, what the co-regulatory proteins are recruited or released in order to decide on the transcriptional activity. In addition, so this whole thing binds to the response element in the DNA of the binding co-regulatory proteins, and also the response element that binds the receptors as homodimers, heterodimers as monomers, can in addition act as allosteric modulator. So the response element in the DNA itself can also influence the whole complex. Then as a third factor, you have post-translational modification of the receptor that can influence the activity. Other transcription factors can bind, so the complex can be extended. And then as a final factor, you have also to consider the individual availability of your cell regarding co-regulatory proteins. So not every cell has the same pattern of co-regulatory proteins. And this can also influence the situation that in some cells, for example, the nuclear receptor works differently as in another one because you have a different pattern of regulatory proteins expressed. So this makes the whole story extremely complicated. And this is probably also the reason why pharmaceutical industry does not like these receptors anymore, because it's more or less a try and error thing, and you cannot really you have no possibility so far to predict what's happening with a specific ligand you design, for example, in the computer. Maybe in the future with AI it might be possible, but I think even then you would need much more data to build here a model. On the other side, although it's so complicated in the past, I mean, these receptors had been pretty much successful. So I found here a paper in Nature Reviews Drug Discovery from 2017, and they tried to find so which targets had been the most privileged over the last years. So what privileged target families can be identified? Of course, I mean, G protein coupled receptors have been very successful, are still very successful. Ion channels, kinases, but also nuclear receptors have been very successful. So these 4 privileged target families, as they call them, make up the targets for 70% of all small molecule drugs available to the date where they did the analysis. So it was, so nuclear receptors had been very successful. I mean, in the beginning, much more. You see, they also show it in certain time frames. So recently there was not much research here, not much development occurring, obviously, but in the past it was very successful. And if you look at this graph they showed in this paper, it was even more interesting because it compares on the one side in blue the percentage of compounds in Campbell, so which means compounds that are identified as biologically active, and in orange the percentage of drugs, meaning if the orange bar is much higher than the blue bar, it means that the target was very successful because most of the bioactive compounds made it into drugs. And if the blue bar is very high and the orange bar very low, that means that you have a lot of biologically active compounds, but very few made it into drugs. So, and you see that also here nuclear receptors had been extremely successful compared, for example, to kinases that are not very successful in the end. Also, there, of course, there had been successful compounds in the market. But I mean, you needed to develop much more compounds in order to, bring them successful on the market. So you can say that nuclear receptors, when you developed your compounds, ligands, then they paid really off because you brought a lot of compounds into the market. So what could be the reason then that in the last, let's say, maybe decades even, the work on nuclear receptors, but was not so successful or a bit disappointing for big pharma? I was thinking about the example of the peroxisome proliferator activated receptor gamma. Because this nuclear receptor is considered as a metabolic sensor regulating adipocyte differentiation, glucose homeostasis, and inflammation. So it was a perfect candidate actually to develop compounds for type 2 diabetes. And so the companies did, so they developed troclitazone, rosiglitazone, pioglitazone. Troclitazone very early left the market because of hepatotoxicity. Rosiglitazone had showed cardiovascular events, and it was also taken from the market. So the only compound I think that it can be or is available in some countries still is pioglitazone, but it is also thought to promote bladder cancer. All of these glitazones have side effects like fluid retention, weight gain, and loss of bone mineral density, all things you don't want to have as side effect, especially not weight gain if you have type 2 diabetes. So what could be the reason that these compounds more or less failed? The natural ligands you find for these receptors are fatty acids and eicosanoids that bind with very low affinity to these receptors. And a colleague of mine in Munich, Daniel Merck, here I give you this source here, he showed even that also thyroid hormones and vitamin E metabolites can bind endogenously to this receptor. And he clearly showed that there are really huge difference between these natural ligands and the ligands pharmaceutical industry developed. So that they differ in binding affinity, in the binding mode, the ligand binding pocket. They are normally much weaker active, so they are acting mainly as partial agonists. So my guess would be if big pharma would have learned in the beginning how natural ligands bind, maybe they would have been better off designing ligands that are, for example, have a lower binding affinity, act more as partial agonists. So normally we think we need a perfect ligand, a perfect agonist or antagonist for a specific receptor, and that's working perfectly. But sometimes it can be too much. I also liked this picture very much when I've seen it once. This is a summary I took from a paper published in 2020, and it shows or gives you an overview what are the natural ligands, the endogenous ligands for these nuclear receptors. And as mentioned already, for the PPARs, mainly fatty acids. So metabolites from the fatty acid metabolism are ligands for PPARs, for these peroxisome proliferator activated receptors. There are 3 different ones: PPAR alpha, PPAR gamma, and PPAR delta. And many nuclear receptors bind compounds that come from the isoprene metabolism. So there are, for example, here you have all the steroid hormones that bind to the steroid hormone receptors, but also a lot of cholesterol metabolic intermediates or metabolites are ligands for the liver X receptors, for example, for the FXR receptor. Vitamin D has an own nuclear receptor, the vitamin D receptor. The ROR, which I'm going to talk about later on, binds cholesterol metabolites. So many nuclear receptors have ligands or bind ligands that come out of the isoprene metabolism. And if you look on this picture, every natural product chemist would say, oh, wow, yeah, I think then we should test more terpenoid compounds, for example, from plants or from wherever. So I think this is pretty obvious here if you look in this picture. So going back to the nuclear receptors, I promised already that I would like to show you the different type of nuclear receptors. I explained already how the steroid receptors work. They are classified as the type 1 nuclear receptors. The heterodimers that are built heterodimers with the retinoic X receptor, which are about 1/3 of all nuclear receptors. They are classified as the type 2 nuclear receptors. And then there are a group of nuclear receptors that are classified as type 3 nuclear receptors that act as dimers also. And very often the ligands are not identified yet. Maybe they don't even have ligands, and therefore they are called orphan receptors. So always when you don't know the endogenous ligand, they are called orphan receptors. And the type 4 nuclear receptors, they bind as monomers. In total, we have 48 nuclear receptors in humans. And I would like now to focus with you a little bit on this ROR receptor. Before we come to ROR, I just would like to show you this picture here. So on the right side, you see basically how such a heterodimer binds here to the DNA. In green, you see here a co-regulatory protein. And then agonists, antagonists, inverse agonists can bind. And so if, for example, you have an agonist to this nuclear receptor, then in the end, it leads to an increase in target gene expression. If you have an antagonist, then you block more or less target gene expression. And if you have an inverse agonist, you can see this also on the left. This is only possible if your receptor is constitutively active, so that it has a basal activity. Then you can have inverse agonists, so that the ligand binds and the activity goes down. So it does not just block the activity, but the activity goes down. So the constitutive activity is inhibited, and so the whole activity goes down. I just explained this because I use this inverse agonist term now because we are looking for inverse agonists of ROR. So ROR are classified as type 4 nuclear receptors. They act as monomers, it's mentioned already. They are constitutively active. That's the reason why we can look for inverse agonists. They are not yet de-orphanized, so they still belong to the so-called orphan receptors. So there is no general agreement on an endogenous ligand for this receptor, but it's known that cholesterol biosynthetic intermediates and also oxycholesterol, so oxidized cholesterol metabolites can act as ligands for this receptor. And these cholesterol metabolites, they act mainly as agonists. The name comes from, so RA stands for RA-related orphan receptor. As always, the nomenclature is quite complicated. So RA is another nuclear receptor, that's the so-called retinoic acid receptor. And since RA has some similarities with this nuclear receptor RA, it's called RA-related orphan receptor. And orphan explained already because there's still no endogenous ligand that the scientific community could agree on. So the name, so the abbreviation RA stands for RA-related orphan receptor. And there are three different isotypes, alpha, beta, gamma. And from gamma, we have even two different isoforms. So for alpha, beta, and gamma, there are three different genes. And gamma T, they come from the same gene. And the only difference is that for gamma T has a shorter N terminus compared to Rho gamma, and it is also exclusively expressed in lymphatic tissue, which makes it a very, very interesting target because it has a very prominent role in lymphatic tissue because it's the key transcription factor here that is responsible for the differentiation of naive T cells into Th17 cells. And once these T cells are differentiated in Th17 cells, they release cytokines like IL-22 and IL-17. So we have here two different IL-17 cytokines, IL-17A and F. And these cytokines are very important to activate epithelial cells and to recruit neutrophils. How does this work? I mean, these cytokines, for example, stimulate epithelial cells in a way that they produce more antimicrobial peptides that directly kill or inhibit the growth of bacteria that are attached to the epithelium. Or they stimulate also the turnover of the epithelial cells so that they divide more often. And by this division, they can also shed bacterial colonization. And they stimulate also stroma cells, myeloid cells to produce the granulocyte colony stimulating factor, which promotes the production of neutrophils in the bone marrow. So the production of neutrophils is stimulated. And also they can help to recruit neutrophils. And yeah, so there's also an increased recruitment of the Th17 cells to the site of infection. So this Rho gamma T has therefore a very important function in defense mechanisms on the epithelium, meaning in the gut, also on skin and so on. But I mean, of course, they are very important. But if you have a problem here, then autoimmune diseases can develop, like psoriasis, for example, because you have here the problems on your skin. Also, in multiple sclerosis, they can the Th17 response can play a role. But also in inflammatory bowel disease, where you have again the epithelium inflammation and so on. So as mentioned already, cholesterol derivatives can act as ligands for these Rho receptors for Rho gamma T, and they normally are activating, so they work more as agonists. And what is also very interesting that, I mean, cholesterol derivatives can also be transformed to bile acids, and it was just recently a huge topic. I give you here also a Nature paper from the year 2022 that they showed that specific secondary bile acid metabolites that are generated by the metabolome in the gut can act as antagonists or inverse agonists of this nuclear receptor. So there seems to be a delicate balance between cholesterol derivatives and bile acid derivatives in terms of activation and suppression of this nuclear receptor. Over the years, also synthetic derivatives had been generated, but so far none reached really phase 3 clinical trials, mainly due to lack of efficacy, sometimes also for safety issues. And the first natural product that was discovered here as ligand was digoxin. When the doctoral student, that is in the meantime now a postdoc in my group, started to work on this topic, the first COVID lockdown just started. So I told him, maybe it's good to invest the time into a review. And so he did. He tried to find out, what natural products are known already as modulators or ligands for these raw receptors. And he found several here that can bind and work as agonists or inverse agonists. So they bind to the ligand binding domain, and then the transcription factor, this DNA binding domain, binds to the response element. And then target genes are regulated, suppressed, or transcribed. And the influence can be manifold. So especially ROR gamma, not ROR gamma T, but ROR gamma has an influence also on the circadian rhythm. As mentioned already, especially ROR gamma T has a very important influence on the balance of the Th17 response versus Treg cells, which are more anti-inflammatory. But also other systems can be influenced, for example, metabolism. So our target gene is, for example, glucose 6-phosphatase. And also IGF-B alpha is a target gene, so it can also have influence in inflammation. And since NF-κB also has influence on proliferation, also on proliferation, cell proliferation. Yeah, and when the lockdown was over, Patrick, the person picture shown here, came back to the lab, and what he did, he just screened the aforementioned database, the DNTA database we have that was optimized on inflammation. And you see, he found quite some hits, but you have to see that here he found activity at the 10 micromolar concentration and here 1 micromolar. So you can see that the terpenoids had been most successful in generating hits compared to other classes. So here is the control, and here is a positive control. So this was the benchmark more or less, and you can see that he had several inhibitors, but here of course he used just 1 micromolar for the positive control, and here 10. But for the terpenoids, 3 terpenoids, he found also quite interesting compounds. So this shows you the assay we are using, the so-called luciferase assay. We are using here 2 different types, just that you know, because I mentioned them now for our data. The one is the GAL4 luciferase assay, and the other one is called full-length luciferase assay. So the GAL4 luciferase assay looks like this. You have the ligand binding domain of interest. So if you're looking for ROR, ligands that bind to the ROR ligand binding domain, we use the ROR ligand binding domain. And this is coupled to the DNA binding domain of a yeast transcription factor. It's called GAL4. And with this, the cells are transfected, and they're transfected also with a luciferase reporter gene that is regulated by a promoter that contains a response element where this GAL4 DNA binding domain binds. So with this assay, you can simply measure whether a ligand really binds to the ligand binding domain of your nuclear receptor, because once it is bound, you will have activation of the gene transcription of the luciferase reporter gene, and you can measure this in the end by the luminescence of the protein that is expressed. So that gives you very clear results because it's a yeast system in a mammalian system included, so you have no crosstalks whatsoever in the cell. So you can simply say, okay, my ligand binds to the ligand binding domain, that's it. And on the right side, you see a little bit more complex system because we transfect here the real nuclear receptor with the full length, so also with the real DNA binding domain that binds to its natural response element that is coupled to the luciferase reporter gene. So, of course, since this is a more natural system in the cell, you have the interaction more probably with other factors in the cell. For example, also if you measure type 2 nuclear receptors, you see probably different results, whether you transfect also in addition RXR or not. This was the first paper we published on ROA. We revisited here a very, very well-known natural product, diosgenin. It's very well known also because it's used very often as starting material for the production of steroid hormones. You can also buy in the internet yams wurzel, so it's sold also as whatever nutraceutical. And we found that indeed this diosgenin, so the aglycon of the main saponins here in this no, I don't know the English word now about Jamswurzel. This drug is a very, very good inverse agonist of ROA. So you can see here the individual bars in green. On the right, you can see the fitted curve. Here in the ROA gamma GAL4 luciferase assay, and on the other side of the full-length ROA gamma luciferase assay. And you see the IC50 values we here found had been in the same range, so about 2 micromolar, I think, which is quite good for a pure natural product. And we also tried to test the original saponins like dioscine and bromdioscine, but as you can expect, they had been very cytotoxic. So for dioscine, we could only test up to 1 micromolar because it was so cytotoxic. And protodioscine, we tested up to 15 micromolar, but we couldn't find any activity here. So it's really an activity of the aglycon of the diosgenin. Here again, the positive control. We tested then also this diosgenin on a variety of other nuclear receptors. We have models in our lab. It was active also on ROA alpha, so a second isotype, which is very similar to ROA gamma. And we found here also an IC50 value in a very similar range. But on the other nuclear receptors, it was inactive. So especially interesting would be here, for example, FXR and LXR, because they bind also steroids or like bile acid derivatives, cholesterol derivatives. And since this compound had been quite active in our luciferase reporter gene assays, we tried then also to find or to measure an effect on gene expression level. So we choose here Chuka T cells that had been transgenially transfected, also with ROA gamma. And here looked on via qPCR on IL-10, so interleukin 17A, and you see there was a dose-dependent inhibition. In the range from 3 to 50 micromolar. And we also looked in HepG2 cells on another gene, the glucose-6-phosphatase, and found also here dose-dependent inhibition of this gene expression. Colleagues of us also did some docking studies, so their final conclusion was that the binding occurs more via hydrophobic interaction, but here the hydroxyl group on the C3 position interacted also with some amino acid residues. It was more the anchor here, anchor molecule. Why did we come to dioskinone? Actually, it was not just a complete random story, but there was a paper before here published in 2010, and they actually have shown already that dioskinine in a murine model of multiple sclerosis, the so-called experimental autoimmune encephalomyelitis, showed activity. So it reduced neuronal inflammation and demyelinization. And what they found, I mean, they did not look on ROR as a nuclear receptor, but then dioskinine binds to the nuclear receptors. They just checked whether ROR gamma is, or ROR alpha is differently expressed. And they found that there was an effect on the STAT3 signaling and that the expression of ROR gamma and ROR alpha went down. So we added to this data previously published the fact now that dioskinine acts as a direct binder of the nuclear receptor, as an inverse agonist of ROR alpha and ROR gamma. So the most interesting compound we had been working on recently is primolagenine A. So far, there's only a preprint published because the original journal we sent it to takes a while to, it's revised already, but still we have no answer. They take months to answer. But you can see or find it as a preprint. Here had been also quite many people involved because it was a very interesting story. Because we found that this compound is indeed extremely active on ROR gamma. So we found here in our luciferase reporter systems, in the GAL4 system and also in the full-length system, IC50 values at the level of 100 nanomolar, which is for pure natural product extremely potent. And in addition, we found that on all these nuclear receptors here on the right, it was inactive. So we tested them on GAL4 acids. So it was also very specific. And we also found no cytotoxicity, which we showed in resacurine conversion assays. So we have been very excited about this finding. And I mean, although it was known that other triterpenoids are also active on ROR, primolagenine A was very much more potent. So here we directly also compared it to oleanolic acid, which was described already as ligand for ROR. And in addition, it's much more specific. For example, oleanolic acid is also reported to bind to FXR, LXR, RXR, and PPAR gamma. So I think it's a very exceptional compound, what we found here. The problem was that this compound is not commercially available, so we had to motivate colleagues from the phytochemistry lab, so Judith Rolling and Ulrike Krieke, also to isolate more for us. So we also accompanied also the isolation process here with our assays. And we could convince also a chemist, Nina Schützenmeister and her doctoral student, to synthesize primolagenine A. So we had two different sources that assured them that we could continue with our work on this compound. And also Nina Schützenmeister's group, she is now working also on derivatives that might be even better than the original compound. We found, so we did again the same gene expression experiments in Chuka T cells and HepG2 cells. And you can see here that in HepG2 cells, looking on the glucose-6-phosphatase, it worked quite nicely, dose-dependently. And you see that we came really in the range, concentration range also of the positive control here. In Chucker T cells, it was less convincing because it inhibited, but it wasn't really dose dependent. And Patrick said that it was always a big problem to get reliable results in Chucker T cells. So he created an own cell line, a cell line where he chose a murine model of T cells, the so-called EL4 cells. And he created stable transfected cells that express that have a stable bicystronic mRNA. So on the same construct, murine raw gamma and GFP. So this helps in the end to sort the cells via flow cytometry. And so he sorted the cells until he had stable cells. And so these cells now express stably murine raw gamma. And of course, from time to time, we have to resort them again, but they work now quite nicely. And you see he looked here on the effect of PGA on the expression of IL-17A and IL-17F and the receptor for IL-23, which is very important upstream cytokine also. And when the T cells, Th17 cells are activated, they express higher levels of this receptor to maintain the whole inflammatory process. So it's important also to have a look on this IL-23 receptor. And you see here in this system, we had also so our compound was equally active as the positive control, and the effects had been dose dependent. We then try to find out whether our compound also works in primary cells. So we established here a collaboration with the Medical University of Vienna because they had transgenic mice here that express, they are called the OT2 mouse because they express stably T cells that have a T cell receptor that recognizes when they are presented by the MH2 ovalbumin. So what you can do is you take their spleen, mesh them, so you have a total spherocyte culture. But from here you can, with ovalbumin and a specific cytokine cocktail, drive the differentiation into Th17 cells because, for example, the dendritic cells then will present ovalbumin to the naive CD4 plus T cells, and then they differentiate to Th17 cells, which produce then IL-17, IL-22, and you can sort them then also by flow cytometry. And so he measured here the differentiation of these Th17s, and you can see that with the positive control, but also with PGA, the IL-17 expression goes significantly down. I mean, here he had 1 micromolar, here he used 10 micromolar, but if you look here on the graph with the dose response, you see that 1 micromolar molar PGA was equally effective than 1 micromolar of the positive control. So it was really very convincing. And then Patrick himself tried to establish a model where he can also measure activity on primary human cells. It was it took up quite some time to establish this model. So he took whole blood from the Red Cross, isolated this polymorph nuclear monocytic cells, PBMCs. I forgot. Anyway, so these are again a cell mixture. We can isolate naive T cells by removing all other cells with magnetic beads. so that you have pure culture of naive T cells, and then you can activate them again under specific conditions to Th17 cells. And again, he set up here an experiment, and he could show also that here with these human primary cells, there was a dose-dependent inhibition of Th17 differentiation. And again, very comparable also with the positive control. It seems even here as if our compound was more active. And then in the very last experiment, he also tried to see whether his compound is able to work on already differentiated cells. So again, with colleagues from the Medical University of Vienna, he sorted Th17 cells and reactivated them under specific conditions, and then treated them with the compounds. And also here he found a reduction of IL-17A positive cells, again, very equally active to the positive control. So in summary, I think I hopefully could show you that we found with a pure natural product, the PGA potent inverse agonist for Rho gamma, especially here Rho gamma T we looked at. It inhibits the differentiation of murine and human CD4 positive lymphocytes into Th17 cells. It is specific for Rho gamma, and as I mentioned already, we would like to do more structure-activity relationships by testing other natural related ligands and maybe also new structural analogs synthesized by synthetic groups. And yeah, and I think I hope I could show you that natural products are very useful guides to identify at least two compounds that can act as ligands for nuclear receptors. And yeah, let's see how this story leads us to. We are still working on it, and I would like to thank the whole group and collaboration partners. And as I mentioned already, the main student, now postdoc in my group that were working on this project is Patrick Schwartz. This is this guy here. And thank you very much for your patience and for listening to my talk.
Okay, Professor, thank you for your exciting talk, such a huge scientific production that was presented here, and very important results. We have a break for some questions from the students, if someone would like to ask something related to your presentation. But I would have a question that it deals more with the methodology of work. I have seen that you work mostly with isolated compounds. And when you work with plant extracts, do you also consider to work with complex matrix and try to fractionate and try to find the most active compound, or you go?
Yeah, we do this also. But when it goes more into some mechanistic studies, it makes no sense to work with extracts anymore. But of course, I mean, to identify active ingredients of mixtures, of course, we offer this to our colleagues. Yeah.
Okay. Something that you have showed that a result of the screening for PPAR agonistics or antagonistics, and as a result, the three terpenoids were the most active compounds. And what could come next? Do you make in vivo models to apply this effect, this in vitro effect for an in vivo model for a specific disease?
Yeah, to be honest, yes, we would like to do this, but we need collaboration partners for this. It's not so easy, to be honest, to convince scientists from the medical university here also to collaborate on this, because they are always busy, of course. Yeah, but this, of course, would be great to have in the end also in vivo data. I mean, it could be in the end that the compound, the natural product, has a drawback in the sense that it's not very good bioavailable. I mean, triterpenoids, I think, are very well known for not being highly bioavailable. So we would expect this, that it's not very highly bioavailable. But on the other hand, I think that also a chance for compounds that are not so highly bioavailable would be a local activity in the gut, because a lot of receptors you can find there, because you have, I mean, the immune system is mainly on sites where you have the biggest entry. This is the gut, the lung. So there, you might even, influence the immune system in the gut locally. This might be also a thought we have, but it's of course very difficult to show this. You would need to show this in vivo, and yeah. What would be very interesting.
Okay. I was curious about this last compound that you showed the activity. It was called PGA, or it's a triterpenoid that you have seen that it has an activity on ROR as an inverse agonist. Which is the source of this compound?
Which one do you think? You mean this one?
Yeah.
Oleanolic acid?
No, the other compound that was preprint the article. This one.
This one?
The next.
And what is your question?
No, the next one. It was in an article that is preprint. No, this is the.
I did not get yet the question. Sorry.
You showed a compound that was a triterpenoid that has an activity on ROR receptor, but a very potent activity. But I was not able to look for this plant source. Which is the plant source? It seems to me that it was a syrup or something like that. I think the next slide. The next. This one.
This one?
Yeah.
But this is the piperacillin A I was talking about.
Yes.
So this is just, you mean the title?
Which is the plant source of this compound? Was it isolated from a specific plant?
Yeah, it comes from Primula.
Ah, okay.
So from what we call primul. So it's a very common plant, what you use actually in cough syrups.
Ah, okay. This is not a very common plant here in Brazil. So that's...
So you can find it everywhere here in the springtime.
There's a question here from a student. Is there a possibility to check if two or more compounds could have synergistic activities?
I mean, of course, you can test this, yeah. Maybe. I mean, you have then simply to combine the compounds, yeah.
Yes, combining compounds, maybe she was referring to. Well, okay, Professor, we will not take more your time, your busy schedule. So, vielen Dank for your presentation. Guten Arbeit.
You're very welcome.
And thank you for the opportunity to have you here talking to us.
Thank you very much. Have a great day. Ours is over.